Within our platform, it's a base system of using a freshly cultured neuron that you can take any disease gene that is expressed in humans that drives the disease process. So you can replicate that within these neurons, and it's ideal if you have a dominant inherited gene, but any also recessive genes will work too. So it would work for basically any human disease that has a genetic component. So you take that human disease gene, put it in the Drosophila neurons, and as long as there's a robust effect in the neurite morphology, then you have a platform for screening drugs. Very early in the technology, my co-founder, Joost Schulte, who's now Chief Scientific Officer of the company, he came to Troy Littleton's lab at the Picower Institute of MIT to work on the Huntington's disease model. So Joost came out of in vivo Pharmaceuticals to work in Troy's lab because at that time the Huntington's disease model that Troy was using was the best one that expressed the greatest portion of the human disease gene, so a larger gene fragment than any other model system. This system was in vivo, so in whole Drosophila animals. And so you couldn't really do drug discovery in whole animals. It takes too long, it's very expensive. We needed to miniaturize it. So the solution was to take this model expressing the human disease gene that has a causal role in the disease and create fresh neuronal cultures. So if you create fresh neuronal cultures out of this model, you can see that the neurons are dystrophic. They look funny. They don't develop as normal cells do. So it's a very strong, robust assay. You can tell clear differences between the freshly prepared neurons expressing the human disease gene versus neurons that don't carry the human disease gene. We wanted to find compounds that would reverse physical properties of the neurons expressing the disease gene, make them look normal. And at the same time, we were also tracking the position of the human disease protein. So the gene that encodes Huntington, where does that Huntington protein localize? To do large-scale screening, what we did was we made these large cell cultures, like volumes of large cell cultures. We plated them onto 384 well plates, so a plastic tissue culture plate with 384 wells. And we then used screening facilities that had compound libraries that had thousands of compounds that could be applied with robotics onto these 384 well plates. So every well in the 384 well plate would have a different test compound. So with these robotic systems, we then collected all the images of the cell cultures. So we tracked the neuron morphologies with a green fluorescent protein and the human mutant disease protein with a red fluorescent protein. And then we did automated image analysis to find our hit compounds. So through that, we found 2 structural classes of compounds that reverted the disease morphology to the wild type healthy neuron. And we also found a novel gene target that suppressed the toxicity of the mutant protein. Also, we had optimized the platform at the Broad Institute as well, using their robotics. But the actual pilot screen that we ran was conducted at the ICCBL across the street, across the river. Alzheimer's is more complex because there's multiple disease genes involved in that. But our ultimate goal is to take multiple disease genes, so do a drug screen in a Parkinson's model. Take that same compound library and test it on a Huntington's model. Take the same compound library and test it on an Alzheimer's model. Then look across those data sets to find compounds that reverse the disease process in each of those diseases. And those hit compounds, the ones that work in all of those models, are likely to be compounds that affect the aging process of the brain. So one thing about neurological or neurodegenerative disorders is they happen with age. And over the course of human brain aging, there are a number of processes that decline. So for example, protein quality control. That's something that is an issue in all of these major neurodegenerative diseases. So if you can find ways to improve protein quality control, these are compounds that are likely to be broadly neuroprotective, so applicable to Alzheimer's, Parkinson's, Huntington's. So as we increase our capacity to do screening within our company, that's our ultimate goal.